Microtraces Incorporated was founded as a corporation in 1961, though its origination came from Anresco Laboratory, where my father, Dr. Sylvan Eisenberg, was the founder and president of both of Anresco and then president of Microtracers, at a planning lunch in San Francisco of the American Serial Chemists. An issue which came up for discussion was quick analyses for different animal drugs added to animal feeds in the 1950s, including nicarbazine for poultry. My father said we could talk all week about such quick tests, and they only had a 2 or 3 hour session. And my father said, why not talk about simple, easy to detect tracers that could be used to code the presence of an animal drug in animal feeds? People looked at my father and said, do you know anyone who's an expert in that? My dad said no. Said, then you talk. And that was the beginning of Microtracers. Thank you for being interested in the subject of validating mixing and verifying the mixing of formula animal feeds, and especially in interpretation of results for particle counts as compared with analytical data generated from manganese and other ingredients in feeds that are commonly used to validate mixing and verifying mixing of feeds. The simplest microtraces that are used widely now around the world are Microtracers F, which are simply colored iron particles with a specified count of 25,000 particles per gram. First, a couple of related issues, not directly related to the interpretation of the results, but critical as a foundation for interpretation of data. In running any mixer test, one must consider the addition of the tracer to the feed, the formulation level of the tracer, which for Microtracers F is commonly 50 grams per ton of feed, and manganese, which is commonly formulated at 100 grams per ton. And adding whatever tracer at a micro level is really important, because the data is being used to support the ability to mix drugs that are oftentimes added at low ppm levels. And testing for something like salt, sodium chloride, is not adequate, because in the United States it's commonly added at 20,000 ppm, 2% of the formula. It's not adequate, not accepted in the European Union. Both tracers, whatever tracers are added, should be added directly into the mixer, and so the addition of the tracer is critical. The next issue, which is critical to performing any mixer validation trial, is the sampling of the feed. Samples must be grab samples, not composites in any way. If one is validating the mixing equipment, then it's ideal to take samples from within the mixer, to use these to support the engineering of the design of the mixer. Commonly, though, people take samples from the discharge of the mixer. In any event, the sample should be taken as near the mixer and the manufacturing system as possible, because otherwise feed from other batches of feed not containing the tracer can commingle with the test feed and thereby throw the results off, and also is a possibility of segregation of feed even before samples are taken, especially of mineral premixes. It is reasonable to assume that if the sampling conditions are demanding, then the mixing will also be adequate for macro ingredients and other ingredients which are added to the feeds at higher levels. So we try to test mixing under severe conditions to support the quality of the mixing under all conditions. Another issue that needs to be considered in performing a mixer validation test is that not all feeds are the same. Feeds that with larger particle size in general will take longer to mix than feeds with a finer particle size, and this is supported by research performed by Tech France and by Kansas State University in the United States. Further, Segregation can be a very real issue, especially with mineral premixes, where ingredients may be of different particle size, and samples may segregate even before the samples are taken. And you can see this by taking a sample of a mineral premix, putting it in a plastic bag, shaking up and down, and you'll see fine material coming to the bottom of the bag and larger particles floating to the top. So segregation is something that has to be considered in running a mixture test. The next issue that has to be considered in performing a mixture validation trial is the analytical method used to determine the tracer that's being used as an indicator for all the other ingredients in the feed. The microtracer's F particle counts are generated by isolating the tracer particles magnetically from feed samples and then sprinkling them onto a large filter paper, developing colored spots that one can then count. In determining manganese, which is a commonly used tracer, it's determined analytically by atomic absorption spectroscopy and requiring, or ICP-MS, either of which require a skilled analyst. The microtracer particle count analysis requires some skill, but not a great deal. Now we get to the interpretation of mixture validation test results. First, for microtracer F particle counts. Microtracer F particle counts are interpreted using the Poisson statistic, which is accepted by statisticians all over the world for interpreting particle counts. The critical property of the Poisson statistic is that if you take an infinite number of samples and you will get a standard deviation for a series of counts equal to the square root of your average count. Based on the Poisson statistic, if one counts 100 particles per sample, one will find the standard deviation to be 10 and the coefficient of variation to be 10% if the mixing is perfect and you took an infinite number of samples. If you wanted a CV of 5%, this can be achieved, but you have to count 400 particles per sample. Then the standard deviation would be the square root of 400, or 20, and your coefficient of variation then would be 20 divided by 400, or 5%. This is just the way, this is inherent to the Poisson particle statistic. But one does not analyze an infinite number of samples in a mixture validation test. One may analyze 10 or 20 samples. And if one is looking for a CV of 10%, the question becomes, if you find a 12% CV, does this represent a statistically significant deviation, or is it merely random noise that can be accepted as still evidencing a complete mix? One employs the chi-squared test, which is linked to the Poisson statistic, to determine whether the trial results, if they yield a coefficient of variation higher than what was predicted by the Poisson, whether this deviation has any statistical significance or not. If the trial results yield a chance probability based on the chi-squared test greater than 5%, this means that in more than 5% of the trials of a perfectly mixed feed, one would find results that were more variable than were found in the trial batch, and therefore the data is not significantly excessively variable to support even a conclusion that the mix is probably incomplete. Since it's not probably incomplete, one accepts it as being complete. If one finds results that would occur in less than 5% of the trials of a perfect mix, one considers the mix probably incomplete. And if one finds trial results that would occur by chance in fewer than 1% of trials of a perfect mix, one judges the mix incomplete because the difference is statistically significant. Conclusions are simple and objective. The inherent analytical method in the microtracer test is about 3%, and that's simply not considered in the interpretation of the results the way we've handled it over the years. Interpretation of manganese and other microingredients as evidence of mixing is based on the normal statistics, simply generating a coefficient of variation for the series of sample results and then interpreting it. There are no criteria for chance probabilities which are clear and objective. These could be generated using Bayesian statistics, but to my knowledge, no one has done so. Simply, if the inherent analytical method variability is 5% based on analysis of one sample, many replicates of one sample, then one simply accepts mixing as being complete. If the inherent analytical method is 5%, then one simply takes a multiple of that, maybe makes it 8% or 10%, but there's no clear statistical justification for making such a criteria. But that's the way the results are interpreted based on the GMP test methods in Europe and generally accepted elsewhere. The GMP plus mixer validation standards developed in Holland and widely used in Europe and around the world are as follows. For microtracers F, results are considered to evidence good homogeneity if there's chance probability higher than 5%. For manganese, results are considered to evidence a complete mix if the coefficient of variation for a series of sample results is 8%, less than 8%. In Brazil, there seems to be a preliminary criteria. The results are considered to evidence good mixing for the manganese if results are less than 10%. For the microtracers F, results are considered to evidence a probably incomplete mix if the chance probability is between 1% and 5%. For manganese, under the GMP plus methods, the coefficient of variation of 8 to 12% is considered evidence a probably incomplete mix. In Brazil, over 10% would evidence a probably incomplete mix. And for microtracers F, a chance probability below 1% would evidence an incomplete mix. And for manganese, a CV over 12% would evidence an incomplete mix under the GMP plus. And in Brazil, over 10% would evidence an incomplete mix for the manganese. An issue of concern to many feed manufacturers and feed regulators is the issue of the sensitivity of the method. And for microtracers F, if one counts 100 particles per sample and analyzes 10 samples and finds a C of E of 14.9% when expecting 10%, this would occur in less than 1 in 100 trials of a perfect mix, and the mix is judged incomplete. Therefore, an excess variability of 4.9%, making no allowance for analytical errors, leads to a judgment the mix is incomplete. For manganese under the GMP plus standards, the difference between acceptance of a mix as being complete, yielding a CV less than 8%, can be compared with a CV over 12% as evidencing the mix as being incomplete, or a difference of about 4%. So the difference in the excess variability for the microtracers as compared with the manganese is almost the same. On the microtracer F particle counts, for an average count of 100, a perfect result is a 10% CV and yielding a chance probability of 50%. You're never going to get a chance probability at 99%, so 50% is perfect. A chance probability of 5%, evidencing a probably incomplete mix, would generate a CV of 13.1%, and for the incomplete mix, a CV of 14.9%. One can conclude that either microtracers F or manganese can be used rather equivalently in validating feed mixing. in terms of the interpretation of the results. There are other serious issues in interpreting mixture validation results that no one has really studied, not microtracers, not GMP Plus or anyone else. The most important one is the issue of type 1 and type 2 errors. The possibility of judging a mix complete based on the trial results, either from microtracers F or manganese or some other analyte, judging the mix complete when in fact the mix is not complete, or type 2 errors, judging the mix incomplete when in fact the mixing is complete. The way to minimize such errors is by taking and analyzing a larger number of samples, and it's more important to take a larger number of samples than to have more precise numbers for a smaller number of samples. The Poisson limit theorem is just one of the statistical proofs that are utilized in supporting the legitimacy of the Poisson statistic. If you all enjoy integral calculus, you can prove out the legitimacy of the Poisson statistic and also its equivalency to normal statistics when counts are extremely high. Thank you very much for having the patience to listen to this presentation. Please let me know your questions either now or later. We live in the world of the internet. We can communicate very easily, and I look forward to trying to provide whatever assistance I can in the future. Thank you so much.